Hello and welcome everyone to today's live session of week 4. Good evening, good evening. So, in today's session, we are going to talk about. So, if you remember, in the last session, we were talking about antibodies, right? The structure of antibodies and how antibodies variable region is rearranged, right? This was the previous session, and in today's session we will talk about the constant region of the antibodies. Okay, so how the constant region of the antibodies are rearranged and how several classes of antibodies are generated. So we will focus on that. Okay, so if you remember, this is where I ended the previous session also, that we were talking about various classes of antibodies: IgM, IgT, IgG, IgE and IgA. এই শোনো শোনো, এটা অফ করে দাও, এটার আওয়াজ হচ্ছে তো কন্টিনিউয়াসলি। ঠিক ঠিক করে তো আওয়াজ হচ্ছে কি? Okay, so we were talking about these several classes of antibodies, which are IgM, IgT, IgG, IgE and IgA. Now, how are these several classes of antibodies different? They are obviously different by their constant region. So if you look at these constant regions, they are color coded by 5 different colors, and you can see here that these exons, so these are the genes of the antibodies. These are the antibody genes, and you can see here that these exons are exons actually code for several different types of or several different classes of antibodies. And how are these how these exons are differentially coded to generate several classes of antibodies, we will see later. But first, let me tell you that or if you have already gone through the lectures of this week 4, you would also know that IgD and IgM, these are the first types or the first classes of antibodies which are produced, right? So antibodies are the initial antibodies are made of IgD or IgM subtype or IgM-DE or IgM class. And how is that? How is this these antibodies expressed? How are these antibodies expressed? So we will see that IgD and IgM, these exons. So the exons which code for IgD and IgM are they lie on the same mRNA, right? Or they lie on the same gene sequence from where both IgD and IgM are expressed. Now you can see here that in this particular DNA segment, you can see here that these are the exons. These are the exons which code for your c-mu gene. So c-mu basically stands for the constant region of IgM. And what are these boxes? These boxes are the exons, right? And here you can see that this is the DNA, because you can see there are 2 strands, the top strand and the bottom strand. So obviously this denotes the double-stranded DNA molecule. And here you can see that these boxes are the exons and the intervening sequences are the introns, which will be spliced out, right? Now in this particular DNA segment, you can see that there are two types of C exons or constant region exons. One is C mu, which is denoted by this blue color, and other is C delta, which is denoted by this purple color. So these four exons actually code for the constant region of IgD. Alright, and you can see here in this single, when this DNA is transcribed, and when this RNA is produced, the primary mRNA is produced, not the mature mRNA transcript, but the primary transcript. You can see that these all these C mu and C delta exons are transcribed into the RNA, primary mRNA. And then what happens, if the IgM has to be expressed, then IgM is spliced out, or sorry, the IgD is spliced out. That means the C delta regions will be spliced out, or if IgM or IgD has to be expressed, then IgM, the constant regions for the IgM is spliced out. How is this happening? See here, this is the expression of IgM. So if IgM has to be expressed, then the constant region of the IgM has to be expressed. It has to be there in the mRNA. So here you can see that this is one polyadenylation signal. So here there is after these C mu genes, the mRNA is being spliced out and poly A tail is being added. So this final mature mRNA, you can see here, this mature mRNA does not contain these C delta sequences, right? So the C delta sequences are spliced out because it has to be IgM, right? So for expression of IgM, these C delta sequences are spliced out, and you can see that these blue regions are coding for the IgM, the constant region of the IgM, right? And here you can see that this VDJ, so this VDJ recombination that I was talking about in the previous session, this VDJ recombination will take place much earlier. So, and this constant region, the expression of the constant region will take place further. So that is why initially you can see that here is the VDJ is already, the recombination of VDJ has already been taken place, and now the constant regions are being expressed. Okay, so this is how IgM is expressed. Now how IgD is expressed, we will see. That is, here you can see that in this, from the same DNA and from the same RNA, the primary RNA, now you can see that the C mu region, the constant region for IgM, is being deleted, like it is being spliced out, and only the C delta region, this is being expressed, right? Only the C delta region is being expressed, and after this, at this polyadenylation sequence or this polyadenylation signal, this poly A tail is being added, right? So here from between these this RNA, you can see that the C mu regions are being deleted, and the final mature mRNA contains these C delta regions, right? So which will be required for expression of IgD. So you can understand how IgD and IgM are being co-expressed, but you can also see that that the expression of IgD or IgM are mutually exclusive events, right? Either IgD will be expressed or IgM will be expressed. So these are the antibodies that are expressed initially. So they are the initial classes of antibody. So initially, by initially I mean that whenever a pathogen is infecting our body for the first time, then after a few days, if the innate immunity is not being able to tackle the antigen, after a few days this adaptive immune response will start, right? And in the beginning of this adaptive immune response, the first classes of the antibodies which will be produced will be IgT and IgM. Later on, when if this pathogen infects us again, then the memory antibodies, there will be some memory B cells which will be able to recognize that this pathogen has infected us before, and then in that case, in the secondary infection, we can see that other classes of antibodies will also be produced, which will be more effective to eliminate the pathogen. So what are these other classes? That these IgG, IgE, IgA, these are the other classes of the antibodies which will be produced, all right, in case of secondary infection. So now, before going into that, how these other classes of antibodies are produced, one more thing I would like to mention here, that is how these antibodies are secreted out of the cell. So we know that if you remember the B cell receptor structure, we will see that the B cell receptor is actually has a similar structure with immunoglobulins or the antibodies, right? But this B cell receptor is different because this B cell receptor is bound to the membrane of the cell, right? So it has a transmembrane region with which it is bound to the cell membrane. But these immunoglobulins, if it has to be secreted, then obviously it cannot be bound to the cell membrane, or it has to go outside the cell membrane. So how does that happen? So here you can see that in the DNA, here you see that this VDJ region has already been recombined, and now here it is actually showing you only the C mu regions, or the constant mu, the constant regions of the IgM, right? And here you can see two sequences here denoted by yellow, M1 and M2, all right? And also there is another region which is SC. What are these M1, M2? M1 and M2 are the transmembrane region, and also it codes for the cytoplasmic tail of the transmembrane IgM. Okay, so M1 and M2 are the regions which code for the region of these immunoglobulins which span the cell membrane, right? So these are responsible for the transmembrane part of the protein. And what is SC? SC is the secretion coding sequence. So SC is the coding sequence, or the part of the DNA or the gene which will be responsible for the secretion of these immunoglobulins. Now obviously, if the immunoglobulin has to be bound to the membrane, then what happens? Let's see. So these, if it has to be bound to the cell membrane, then these M1, M2 regions, these are transcribed, all right? And so here also you will see, in case of secreted IgM also, these will be transcribed. But later on, by differential RNA processing, either these M1, M2 regions will be present in the mature mRNA, or they will not be present. And depending on that, if these M1 and M2 regions, if they are present in the mature mRNA, then these sequences will code for the transmembrane region of the protein, and thereby, so you can see here this carboxy terminus for transmembrane IgM. Here these yellow regions, so they are coding for the transmembrane region, right? So M1 and M2 are these sequences which are coding for these for this transmembrane region of the IgM, and so therefore this particular immunoglobulin will be bound to the membrane, or it will be transmembrane. Whereas if the immunoglobulin has to be secreted outside the cell, then these M1 and M2 should not be present in the mature mRNA. Here you can see that these M1 and M2 regions are not present in this mature mRNA, right? So how did they disappear? Obviously by splicing, right? Whereas this orange region, which is the secretion coding region or the SC region, is present in this mature mRNA. Now you carefully compare these two mRNAs. This one is for the transmembrane region and this one is for the secreted region, the secreted IgM. So here in this transmembrane IgM, you can see that this secretion coding sequence is spliced out. The secretion coding sequence is not present on the mature mRNA, right? Whereas these M1 and M2 regions are present here, but these M1 and M2 regions are absent in this mature mRNA, whereas the secretion coding region is present, right? So depending on whether the M1 and M2 is present or absent, and also the SC region, the secretion coding region, is present or absent, depending on that, the IgM will either be transmembrane or be secreted, right? So if the secretion coding regions are absent, then it will be secreted. If the secretion coding region is present, then it will be secreted, and the transmembrane regions has to be absent, and vice versa for the transmembrane IgM also. All right. Please let me know if you have understood it or if there is any.
Yes, ma'am, very well.
Okay, thank you. All right. So next, moving on to another recombination event, which is class switch recombination. What is this class switch recombination? This is very similar to your VDJ recombination also, right? So VDJ recombination, we were learning in the last week. What is it for? What does it help in? Or which part of the antibody?
Variable region.
Yes, the recombination of the variable region. Whereas this class switch recombination is for the constant region of the antibody. This recombination takes place in the constant region. So how does this happen? First of all, in VDJ recombination, we had mentioned one sequence, that is RSS. Anyone remembers what was RSS?
Recombination signal sequence.
Yes, very good. Recombination signal sequence. And this RSS was actually the sequence which was actually upstream to any of these V or D or J segments, right? So RSS was upstream of these VDJ sequences, and we saw that 12 base pair RSS can combine with a 23 base pair RSS, and this was this 12-23 rule, right? And then if these sequences combine together, they will help in the recombination event. Here also, in case of class switch recombination, we will see there is another type of sequence which is very similar to RSS, that is this switch signal. Here, this diagram is little complicated, but you can see that these the switch signals are denoted by S. So S mu is one switch signal, S gamma 3 is another switch signal. Here you can see S alpha 1 is another switch signal, right? What are these S alpha 1, mu, these things? These are actually the switch signals which are upstream to particular constant regions of the either IgM or IgG, right? So IgG is Here you can see that before the constant region of C gamma 3, you can see that there is the S gamma 3, which is the switch signal for the constant region, right? So these recombination sequences are present before upstream the constant regions of these in this gene, in this antibody heavy chain gene, right? And the thing that happens here is that this class switch recombination is dependent upon many events. One of them is cytokine signal. So cytokine signal is what? So actually, depending on which cytokine is being produced, it will help to actually express particular types of antibody constant region. Alright, so depending on which cytokine is being produced, the particular type, the corresponding type of antibody class will be produced. So one is cytokine signal for class switch recombination, another is the recombinase, right? So in case of VDJ recombination, what was the recombinase? What was the enzyme? Anyone remembers?
TDT, was it TDT?
No, not TDT. TDT was the enzyme which was actually helping in addition of these P and N nucleotides, rather N nucleotides.
Yes, NP recombination.
Yes. So what was the enzyme which was responsible for VDJ recombination? There were two, like one enzyme having dimers, forming dimers.
Yes, RAG1, RAG2.
RAG1, RAG2. Exactly. RAG1, RAG2. Very good. So RAG1 and RAG2, they form this dimer and they help in this. They are the principal enzymes which help in VDJ recombination. Here, in case of class switch recombination, another enzyme which is AID. Okay, AID is one enzyme which is responsible for this class switch recombination, and this is actually the recombinase, right? Okay. Anyone has any idea what is this full form of AID?
Ma'am, once again, ma'am.
What is this full form of AID? Anyone has any idea?
Actors.
Somebody has replied in the chat box, activation induced cytidine deaminase.
Yes. Oh, actually I didn't check it. Exactly. Very good. This is actually the enzyme which is responsible for this class switch recombination. Activation induced cytidine deaminase. And what is this enzyme? So this enzyme is actually responsible for removal of the cytidine. Okay. And so what it does is, when there is when the DNA forms a bubble kind of structures, when it is not double stranded, when it remains as a single stranded form, so this AID enzyme can come and cleave at this at a point where there is a cytidine actually. So C nucleotide is present, cytosine. So there it can make a cleavage and it can remove this cytidine, and in that place there are other enzymes which will add nucleotides, and this will create a apurinic site, and this will actually induce DNA repair at this point. And this is how this actually recombination event will take place. So I'm not going to give you much details about it, but here you can see one thing, that is, suppose when this IgG has to be expressed, or if the IgA has to be expressed, here you can see, then what happens? So first of all, this cytokine induced expression occurs, right? So here you can see that this is denoting what? This is denoting transcription. This arrow is denoting transcription. So this region is being transcribed, and this AID is the enzyme which is now helping in this recombination event. So you can see here that recombination takes place between the switch signals of the mu and the S alpha 1 region, right? So IgA and IgM. So between the switch signals of IgA, constant region of IgM and IgA. And here you can see that this intervening loop, so it forms a loop kind of a structure like this, and this, so it comes like that, this one, right? Here the switch signals are present, and this intervening loop will be excised, right? So here you can see that this loop is excised out, and then this S mu S alpha 1 recombination is complete, and you can see that this C alpha 1 region is present in this mRNA. So therefore you can see that this IgA will be expressed. Alright, so this is more or less what happens in the class switch recombination. You can read about it more from this very good review that I have gone through, and I have taken this particular figure from this review actually. So you can go through this if you want more information about it, right? So this is the overall process, the class switch recombination, taking the help of AID enzyme, the recombinase here, and the help of the cytokine signals, and also the loop kind of thing. The DNA is being looped out, and it is being the recombination, after the recombination event, the particular Ig class or the immunoglobulin class will be expressed. Alright, so if that is okay, then I'll go to once
I have
more.
a query here.
Yeah, sure.
Could you please repeat about the apurinic site once more?
Yeah, actually, if you go through DNA
In this
repair,
event of recombination, where does this apurinic site become is created?
it happens at the beginning of the recombination actually. So there are several enzymes which are involved in this process. One of the first enzyme is this activation induced cytidine deaminase. So what it does, it actually cleaves off this cytidine, right? And if it cleaves off cytidine from this, suppose this is the DNA strand, it has A, T, G, C, C, A, T, G, this kind of sequence, and what it does is that this AID actually cleaves off this cytidine, so it releases this cytidine, right? And then what happens, it creates this apurinic site, so the purine is absent here, and then there are several enzymes which come into action, and then they actually modify this particular site, so it repairs this particular site, following which this recombination takes place. Okay, so there are actually several enzymes which are involved in this process, and if you want to learn more, I can send you materials in the forum about this, and this review also you can go through. Alright, because I won't have so much of time to explain this whole process to you right now. Is that okay, more or less?
Thank you so much, ma'am. Thank you, ma'am.
Okay, all right. Okay, so now moving on to the next section, which is if these several different types of immunoglobulins are expressed, how are these expressed? So most of the immunoglobulins are of this particular structure, this particular structure, the IgD, IgE. These immunoglobulins are expressed in this particular form, but there are several Igs like the immunoglobulins which are expressed in multimeric forms like IgM and IgA. You can see here this IgM is expressed as a pentamer and IgA is expressed as a dimer. So I have these detailed structures of how pentameric and dimeric IgA looks like. Pentameric IgM, so you can see there are 5 units, right? And they are joined by one polypeptide chain which is called the J chain. Alright, so this is also a polypeptide chain which joins these 5 units of IgM. And in case of IgA also you can find that there is one polypeptide chain, J chain, which joins these 2 units of IgA. Right? And in the last session we were actually discussing one interesting question which was if the multimeric IgM or the pentameric IgM, if it is run in SDS page gel, how many bands you will observe. Okay, and the answer was 3. So here I am just explaining that question once again, that is, if you run this pentameric IgM in SDS page, so of course all these in SDS page this protein will be denatured, right? These multimeric form will denature, and here 3 bands will be observed. So why 3 bands? So of course this individual units of IgM have 2 light chains and 2 heavy chains, right? So they will form 1 band each. Okay, the light chains and the heavy chains, they will form 1 band each. Another band will be formed because of the J chain. Okay, so therefore we can see that in SDS page 3 bands will be generated, right? The pentameric IgM, if you run in the SDS page, it will generate 3 bands. Can you tell me how many bands will be generated if we run the dimeric IgA in SDS page?
In 3 chains.
Yes. So if we run this IgA also, it will also generate 3 bands in SDS page. Acha, can you tell me how many bands will be generated if we run this in native page?
Single band.
Yes, single band. So native page is what? Native page is non-denaturing page. Non-denaturing means what? The SDS treatment is not done for native page, so therefore there will be no denaturation of the protein. And if we run it in the non-denaturing page or native page, it will form 1 band because it will migrate as it is, right? It will not lose its structure. Alright, so this is how IgA and IgM looks like in their multimeric forms. Now moving on to the next segment of today's lecture, that is, I will be discussing a little bit about these MHC class 1 and class 2 molecules. So previously also we discussed some of the details about MHC class 1 and class 2, and you know that MHC is nothing but major histocompatibility complex, right?
Ma'am?
Yes.
Ma'am, once more, ma'am, IgA, IgM.
Yes.
Ma'am, one doubt, ma'am. Ma'am?
Yes.
IgA bond, 3 bond or 1 band?
IgA will also
forming-
produce 3 bands in SDS page. In denaturing page,
IgM is 1.
IgM will also produce 3 bands in SDS page. But in non-denaturing page, it will form only one band. It will show only one band because the protein structure will not be denatured and the chains will not be separated.
Okay.
All right?
Okay.
Okay. All right.
Surasree, can you
Yes.
please elaborate the J chain? Like, what is that? I didn't get any information in lecture also. Like, what is the J chain? Like, what is it made of?
So it is actually a polypeptide chain. So made up of some amino acids.
Is the same one as the VDJ, that J?
No. Very good question you have asked. It's not the same J segment that is present in the variable region. It's not that. It's a separate J chain. Like why it is named as J chain? Because it's joining. Right. That is only the thing. But it is not same.
What is the full form of VDJ? J in VDJ?
That is also joining.
Okay, thank you.
That is also joining, but here since this polypeptide chain is also joining these units of the antibodies, that is why it is also called J chain, because it is also joining, but it is not same with the J of VDJ. Okay, it's not coded by this J segment of the variable region, so it is separate. All right.
So, for our knowledge, like that much details is not required. Like, what is the J chain and
No,
more
you can
details.
actually go through NCBI to look at what is its sequence per se, like amino acid sequence or DNA sequence. You can go through this information through NCBI, if you know.
Yeah.
Yeah, but like other than that, I don't think much information is required about it.
Oh, like it's just some amino acid helps to bind it, the IgM together to make
Yes,
it pentamer.
yes,
So,
yes.
what is the significance of it? Like, why it is forming pentamer rather than like others are not forming, right? So, why IgM is forming
Because
this
there are
kind of
some
Avidity. It is increasing the avidity. There
Yeah.
are 10 antigen binding sites. This is the primary antibody.
Absolutely. It is increasing the avidity, first one point, and like structure wise, if you want to see, like if you want to investigate why is it forming this pentameric structure, that is at the end, like you can see here, these are actually joined at the end of these constant regions, right? So at the ends of the constant regions are joined together. So there are some cysteine rich sequences at the end of these constant regions, which help in this kind of disulfide bond formation, right, which are held together by this J chain. So the rest.
So it will not impact like effector function, like all constant regions are
Exactly.
concentrated.
That is what actually Vandana also mentioned, that it is going to affect the effector function, because it is going to increase the avidity of the antibody, right? So
Yeah.
if there are so this is the IgM is one of the primary antibodies which are going to be produced when a pathogen is infecting our body, right? And through this formation of this particular pentameric structure, this particular pentamer structure can actually hold 10. Oh, I'm very sorry, this should not be the case. It can actually hold 10 antigen molecules together. These are the antigens, right? So by holding to or by binding to 10 antigen molecules together, it can definitely help in better effector function, right? So it can help in clearing the antigen much faster. So which is why this multimeric form of IgM is very essential during the initial course of the infection.
Okay.
All right.
Okay. Thank you.
All right. Okay. Now moving on to the several classes of MHC molecules. Now if we look at the structure of the MHC molecules, the MHC class 1 and class 2, they have very similar structures but slightly different. So here you can see that in case of MHC class 1, there are two chains. One is the alpha chain, which comprises of three segments, alpha 1, 2 and alpha 3, and on the other chain is beta 2 microglobulin. And here in case of MHC class 1, you can see that the alpha chain, that the alpha 3 chain is only bound to the cell membrane, so it only has a transmembrane region. Whereas in case of MHC class 2, you can see there are two chains, alpha and beta chains, and alpha chains have two segments, alpha 1 and alpha 2, and beta chains have two segments, beta 1 and beta 2, whereas both the chains have transmembrane regions, so both are bound to the membrane of the cell, right? And this alpha 1 here, the alpha 1 and alpha 2, these two chains create the, when they come close together, they form this peptide binding cleft where the antigenic peptide will be able to bind, right? And in case of MHC class 2, you can see that the alpha 1 and beta 1, they together form this peptide binding cleft, right? This is the basic structural difference between MHC class 1 and class 2. Now, an interesting difference lies in the binding of these antigenic peptides by these MHC class 1 and class 2. What is this difference? First of all, MHC class 1. So MHC class 1 has a restriction of the length of the antigenic peptide that it can bind. So the restriction is around 8 to 13 amino acid residues. So the antigenic peptide has to be has to lie in between 8 to 13 amino acid long. And why is this required for MHC class 1 and not for MHC class 2? So MHC class 2 does not have any restriction in the length of the antigenic peptide it can bind to, okay? So in case of MHC class 1, you can see, so this is the ribbon structure of MHC class 1, and in between, so, and this is what the peptide binding cleft. So this peptide binding cleft, you can see, is lined by the alpha helices on both sides, okay? And how are these peptides binding to the peptide binding cleft? So there are several interactions which help in this kind of binding, and you can see that in case of MHC class 1, there are several amino acids at the end of these peptide binding cleft, okay? So these amino acids at the end of this peptide binding cleft, so part of the MHC molecule, right, which actually help to bind the antigenic peptide by several types of interactions like ionic interactions or your non-covalent, several non-covalent interactions like hydrogen bond formation and that. So these residues at the two termini of this peptide binding cleft help in binding to the peptide, the antigenic peptide, and these residues are more or less these residues are conserved between several MHC classes. And so suppose like not MHC classes but several MHC molecules, I mean to say, like in case of MHC, so suppose there are two molecules of MHC class 1 which are the two different molecules of MHC class 1, and you will find that most of these end residues, these terminal residues, are conserved so that they can help in binding of the antigenic peptide. And here you can see that this particular peptide is lying such that it can fit into this peptide binding cleft, right? Because and the end are closed by this kind of interactions, right? All right. And what happens in case of MHC class 2? That is, this kind of terminal interactions are not present. So here also there are other interactions. Like what do I mean by other interactions? That is all along the peptide binding cleft. So here you can see these are some of the amino acids which lie along the peptide binding cleft, which are now interacting by hydrogen bond formation or ionic interactions with the antigenic peptide here. Okay, so these kind of interactions along the length of the peptide binding cleft are absent in this case, right? You cannot see here many amino acids interacting with the length along the length of the MHC, right? So this is the major difference between the peptide binding between MHC class 1 and class 2 peptide binding. That is, in case of class 1, the binding mostly happens at the terminal residues, and the terminal residues help to anchor to the antigenic peptide. Whereas in case of MHC class 2, there is no such restriction of a length, because all along the length of the peptide binding cleft there are several residues which are interacting with the MHC class 2 protein, because of which any length of amino acids can be fitted into the peptide binding cleft of MHC class 2. Am I clear in this
Ma'am,
case?
as far as I have a query, I'm sorry to interrupt.
Yeah, sure.
I have a ma'am, in the class it was taught that for the MHC class 2 molecule, the antigen length can be anywhere between 12 to 20 amino acids. It is longer than that of MHC class.
Exactly. So by the term that I'm meaning to say no restriction, it is actually it actually means that it can be greater than 13. So this 13 is a restriction for MHC class 1. But in case of MHC class 2, obviously you can understand that 100 amino acid long peptide cannot fit into this peptide binding cleft, right? So by the term no restriction, I mean to say that it can bind a longer peptide which is like longer than 13 amino acids, right? But if a peptide is longer than that, like if a peptide is like 25 amino acids long, then also this peptide will be processed such that it can bind to the MHC class 2 molecules, and this processing happens in the endoplasmic reticulum, where the MHC class 2 will bind to the antigenic peptide. There only the peptide is processed such that it can bind to the MHC class 2 if it is longer than suppose 20 amino acids or so. All right.
Thank you so much, ma'am. Another query, ma'am.
Yeah, sure.
In MHC class 1, only the end terminal of both the alpha 1 and alpha 2 are interacting, and it was said that these sites.
Not only the termini, there are majorly. There are
and
other amino acids also in the peptide binding cleft, like you can see here, right?
Yes.
Which are also interacting with the antigen binding peptide, the antigenic peptide, but majorly this interaction is governed by these terminal residues.
And these terminal residues are conserved throughout different MHC class 1 molecules.
More or less, because you know that MHC molecules are polymorphic, right? If you have heard of the term. So that means,
yes, ma'am.
This polymorphism means different alleles of several hundreds of alleles of MHC molecules are present in human population, and this polymorphism, these different amino acids, so there will be different amino acids. This polymorphism is actually mainly present in this peptide binding cleft only, so that it gives a selection, like which MHC class 1 molecules will bind to which antigenic peptides. Are you getting my point? Like this polymorphism.
Yes, ma'am. It gives a variety to bind with different kinds of antigens.
Exactly, right.
My query here was, since in the MHC class 2, the antigen throughout the length interacts with both the alpha 1 and the beta 1. So shall we interpret there is conservation throughout the length? The sequences are conserved.
Actually, in GenWay there is one good diagram, but unfortunately I cannot show you right. Or just wait, I can show you once. This particular thing that you're asking is there in this GenWay book actually. let me go to that page once and then show the screen. Okay, right.
Ma'am, don't bother if others have to wait because I'll read Genway and if I have any query, I'll message it to you.
No, I'm just showing it just once. It's okay. You can see this screen, right? Here,
Yes, ma'am.
this diagram is there, and here it is stating that peptides which bind to MHC class 2 molecules, they are variable in length and their anchor residues lie at various distances from the ends of the peptide. So these sequences you see here,
Yes, ma'am.
these actually these residues that are shown in green, these are the residues which bind to the peptide. Okay, and so this is actually the different peptides which bind to the human MHC class 2. These are shown, and you can see that in the antigenic peptide there are several amino acids which help in this kind of binding. Okay, and here you can see, suppose L, phenylalanine, leucine, phenylalanine, isoleucine. So either aromatic amino acids or hydrophobic amino acids. Okay, or here you can see tyrosine, phenylalanine, right? These or there are lysine also. So several types of amino acids which have either the hydrophobic group or the charged groups, they help in this kind of binding interaction. So these amino acids actually determine which particular antigen will bind to which particular MHC class 2 molecule. Got my point?
Yes, ma'am.
All right. You can go through it later. All right. This
It was taught in the class also. I had forgotten about this. Thank you so much, ma'am. Thank you for recalling it.
Yeah. Okay. Right. Okay. Now I'll go back to my original presentation. Okay, so this is more or less the difference between MHC class 1 and class 2 peptide binding. Now, there is also little difference between the MHC class 1 and class 2 when it binds to this CD4 and CD8 co-receptors. So, if you remember that MHC class 2 binds, or MHC class 2 is associated with the CD4 co-receptor, whereas in MHC class 1 is associated with CD8 co-receptor. Now please remember that MHC molecules and CD8, CD4, these are expressed on two different cell types, right? MHCs are expressed on the APCs, the antigen presenting cells, right? Whereas the CD4 or CD8, they are expressed on T cells, right? And how these MHC, different MHC molecules bind to different co-receptors, CD4 or CD8. You can see here there's a difference in structure between CD4 and CD8, and here this, in case of this CD4, you can see that D2 and D1, these are the two domains which help in interaction between the MHC class 2 and CD4, whereas in case of CD8, you can see that CD8 interacts with the these alpha 2 or the alpha 3 region of the MHC by this alpha beta chains of the MHC class 1, of the CD8 molecule, right? So in case of CD4, only D2 and D1 regions are responsible for interaction with MHC class 2, whereas in case of CD8, both the alpha and beta chains, they are responsible for interaction with MHC class 1 molecule. So, is that clear? Anyone having any other doubt?
Absolutely clear, ma'am. Thank you so much.
Okay, thank you. If anyone having any other doubt, or else we will go on to the questions, the question answer.
Yes, I have doubt regarding some basic questions basically, because after this what I came to learn that B or T cell can express this MHC1 as well as MHC2, right? So I want to understand the process. For example, I was thinking that any antigen comes, so for MHC1 or MHC2, first we need to process it, right, inside the cell. Then only it can be expressed on the surface. So
exactly,
they, like B or T cell, play kind of like, it will take the antigen inside, like how macrophage or dendritic cell eats it. Oh, like that it does. Okay. So then it comes to the surface. So now specifically,
so macrophage first of all intakes the not the antigen, the pathogen, right? And then what happens? It presents. Then actually, first of all it intakes, then it chops off this particular pathogen, and so by this chopping of this pathogen, several peptides will be generated, right? And these peptides will be of that of the pathogen, and now the cell will express these particular peptides on the surface. The APCs will express these particular peptides on the surface so that they can be recognized by naive T cells, right?
Okay, so the processing is being done in macrophage or dendritic cells, not inside the B cell.
No, not inside the B cells, but remember that these MHC class 1 molecules, these are expressed on all nucleated cells.
Yeah.
B cell is also one of the antigen presenting cell. It is dendritic cell, macrophage and B cell. So all three are involved in processing of antigen.
Exactly. That is why I'm just going to tell you this thing, that MHC1 is expressed on all nucleated cells. So obviously MHC1 will also be expressed on the B cells as well, right? Only the red blood cells, for example, which does not have a nucleus, right? The mature red blood cells does not have a nucleus. Only on the mature red blood cell this MHC class 1 molecule will not be present. But all the other nucleated cells, they can actually express this MHC molecule, and they can present this antigen by this MHC1 molecule. You understand? So that is why B cell also is one of our antigen presenting cell which can present its antigen by MHC class 1 molecule.
So like those pathogen, only dendritic cell and macrophage like chops off into like...
Yes. Actually what happens, so this is something that
shall I try to attempt this answer?
Yeah, sure, go on.
Ma'am, I would like to explain it in this manner, that MHC class 1 molecules are present in all the body cells. And which cells are reading these MHC class 1 molecules? They are which type of T cells? Those T cells that contain CD8 co-receptors. So these cells are called as cytotoxic cells. So cytotoxic cells are responsible for the killing or lysis of viruses and the tumor cells.
Right.
So this interaction between MHC class 1 and CD8 co-receptor will be only for the immunity against viruses and for tumor against the tumor. Whereas in the MHC class 2, these MHC class 2 molecules are presented by the antigen presenting cells. And the antigen presenting cells are three types: dendritic cells, macrophages, and the B cells. So all these three cells will present these MHC class 2 molecules, and they are interacting with CD4 cells. These are helper T cells, and then helper T cells are playing a role against various types of pathogens such as bacteria, parasites, and I don't know the rest. Thank you so much.
Yeah, very well explained, Vandana. Actually, she explains most of it, but I just want to mention one thing, that is, she clearly explained this, that MHC1 is for the. So there are two types of antigens I mentioned, if you remember, extracellular and intracellular. Okay. So for internal antigen processing, MHC1 molecule is responsible. And for extracellular antigen, MHC2 is responsible.
Like, can you explain this
intracellular? What do I mean by intracellular? Basically, when virus infects, so she mentioned it also that in case of most of the virus infection, MHC class 1 molecule, the antigens are presented by MHC class 1. So what happens in viral infection? So virus is only active when it enters the host cell, right? And when it enters the host cell, it is creating its own proteins. Then these viral proteins are chopped off inside. So there are these so inside the endoplasmic reticulum
of the body cells, say for example hepatocyte.
Exactly. So in all the body cells, whenever there is a viral infection, suppose, so this particular virus has gone inside the cell, and now it is producing its own proteins, and this protein production is taking place in the host cells ER only, right, the endoplasmic reticulum. And in this ER, a part of these proteins will be able to actually join or bind to the MHC class 1 molecules, and then they will be expressed on the surface of these kind of cells, all these types of cells, right, all any nucleated body cells, right. Whereas in case of MHC class 2, what happens, so they are responsible for presenting antigens which are extracellular. And what I mean by extracellular, that is, they are taken up. So they were outside the cell, right, for example bacteria. Okay, they do not need to enter the cell like the viruses, right. So suppose they were outside, and then they are taken up by these antigen presenting cells, and then they are processed inside the phagolysosome, right, so which is the fusion of the phagosome and the lysosome. And then after this processing, these antigenic peptides are presented with the help of MHC class 2 proteins on the surface of these kind of antigen presenting cells.
Okay, let me come back to my original question, which is that so B receptor is actually not chopping that peptides, right? The pathogen.
No.
So okay, so now I know that B cell can be of two types, right? One is they can present MHC class 2, as well as they can present like Ig proteins, right, on the surface. So
no, they are not. That does not necessarily mean that they are of two types. One cell can also present both, like MHC class 2 proteins and also the immunoglobulins.
Ig. Okay. So like B cell on the surface, they actually keeps all these things together.
Exactly.
So whichever got attacked,
and that is what I actually wanted to say before Vandana actually gave her very nice explanation. That is, remember that these MHC proteins are not only presenting the pathogenic peptides. That is the first point that you must remember, everyone. That is, the MHC proteins are always presenting some of our self-peptides as well. And remember, so when we will go through this B cell maturation and T cell maturation processes in much more detail, so you will see that all of these body cells, that the immune cells, the body's immune cells, has to recognize that has to recognize between self and non-self, right? So this was, I think this term was introduced to you in very preliminary immunology classes, that is what is self and what is non-self. So the immune cells has to recognize what is self, what is my own, and therefore it must not react with whatever is self. Okay, otherwise it will produce autoimmunity. So it will produce autoimmune disorders. So if the immune cells has to recognize between the self and non-self, then it has to recognize the self first. So during the maturation of B cells and T cells, we will learn later on that they are recognized, or they are recognized with our own proteins. In the thymus where the T cells are developing, for example, they are presented with our own proteins. And how are they presented? By our own MHC molecules, right? And there, if the T cell reacts with self antigen, okay, then this particular T cell population will be deleted. Okay, this particular.
I think it's being discussed by Agnew Ganguly, Professor.
Yes, sir. Yes. Actually, sir discussed this thing, but he will later on again tell you, or actually give you more details about it. So you understand that all our body cells, they express MHC molecules, and they express our self peptides also in any natural time also, in any normal time when there is no infection also. Okay, and the circulating T cells, when they come across these self antigens, so they understand that this is self antigen and I must not react with it. Okay, so that is it. So it goes by, and so that is what is happening. You must understand this, that in any normal situation, the MHC molecules are presenting peptides, and not only pathogenic peptides. Whenever there is infection, the MHC molecules will present the pathogenic peptides as well, which will be recognized by the T cells and or the circulating whatever the naive cells are there, the naive immune cells. They will recognize that this is pathogenic and this is not myself, right? So that is non-self. So I hope.
So just last one clarification,
yes,
for the intracellular one. So it's inside the cell. So inside the cell, virus is there. So who will do the chopping of the?
That happens in the ER. That happens in this. Like proteasome is also there. You know this proteasome, where the chopping actually it causes this chopping of this peptides,
Like
and then
kind of liposome or something.
No, there are, so you can actually go through this 26S proteasome. You can search for this term. If you have any cell biology, so if you have any cell biology course, or if you have any cell biology book, you can look for it. So this proteasome has a particular structure where these peptides are actually chopped off.
So it will be inside the cell?
Yeah, inside the cell, absolutely.
Okay.
And then these chopped off peptides will associate with the MHC molecules inside the ER, and then they will be presented on the cell.
Okay. And just to clarify, like extracellular means, let's say the bloodstream, or intercellular.
Extracellular means, suppose this bacteria. Most important example, bacteria. So they remain in the extracellular space. They not only they are not certainly they are inside the cells like the viruses, right? So if the bacteria is outside the cell, from the extracellular space, the macrophages, for example, they uptake this bacteria, they lyse this bacteria inside the phagosome, and then inside the phagolysosome, and then these proteins, these antigenic peptides will be chopped off, and then they will be expressed with the help of this MHC class 2 proteins.
Yeah, I just trying to understand like what is extracellular, like bloodstream.
Bloodstream. Yes.
White flow, all these things, right?
And and in the tissue, so there will be the tissue is made up of cells, right? And just outside the cell, there will be. You understand? Like the extracellular space.
Yeah. Okay.
Yes.
Thank you.
Okay. So, any other queries? Yes.
I have doubt from another topic. Would you like to explain, or maybe later?
No, you can ask.
Thank you, ma'am. Ma'am, I would like to ask you, ma'am, can you explain again the diversity of antibody? I'm facing a lot of problem in that topic.
All right. Yeah, so diversity of antibody. I'm just going back once to that particular slide. Yeah, so in our previous classes, we were talking about how different variable regions are produced in several antibodies, right? So the V, D, and J segments, they when they are recombined, so they produce several varieties of the variable region of the antibodies, right? So that is what happens in the variable region. Now antibodies have two regions, the variable regions and the constant regions. Now, so there are several varieties of these variable regions, all right? But are there varieties in the constant region as well? The answer is yes. There are varieties in the constant region, but the variety, the number of classes is obviously much less. That is IgM, IgD, IgG, IgE, and IgA. These are the 5 classes of antibodies which are produced. Now, why are these classes different and how are they different? They are coded by different exons. As you can see in this particular gene segment, there are several exons which code for these different antibody classes. And not only that, these antibody classes are different by the term in term of their effector function, right? So this constant region, previously also I mentioned, this constant region is important for effector function of the antibody. And these several classes of antibodies are actually responsible for different effector functions. So that is one thing. Now coming to how these antibodies, these several classes of antibodies are produced. So I told you when a pathogen is infecting our body, so the first antibody response comes from IgM or IgD. And I told you that these IgM and IgD, these are coded by different sets of exons. C mu codes for the constant region of IgM and C delta codes for the constant region of IgD, right? And by alternative splicing, so either the C mu region will be coded, as you can see this C mu region is coded in the mature mRNA, or the C mu region will be deleted and C delta region will be coded, right? So if the C delta region is coded, it will create IgD, it will express IgD, and if the C mu region is coded, it will express IgM, right? So these are the first initial types of antibodies which are produced to tackle the infection. Later on, during this course of infection, other types of antibodies will be also produced to deal with this antigen in a much more better way, so that to improve the effector function. And these are these IgA or IgD or IgG, and the mostly produced antibody is IgG. Okay, immunoglobulin G is the mostly produced antibody, and here this happens by class switch recombination. So the DNA has the exons of other constant regions of the immunoglobulin as well, but during later courses, during the later phase of infection, what happens that these switch regions are present, the switch signals are present, which actually determine which region will recombine with what. And then suppose if this and what happens by recombination process, this intervening DNA sequence is looped out and it is excised like this, you can see here, and the particular constant region will be expressed. So here you can see that S mu and S alpha 1, there is a recombination between S mu and S alpha 1, and of course because of that this IgA constant region will be produced. So IgA antibodies will be produced, and this way the other antibodies like IgG or IgD, these are also produced. Okay?
Yes, ma'am.
Is that more or less clear with you? The diversity of antibody?
Yes, ma'am. Thank you.
Okay,
yes,
all right,
yes,
all right. All right, any other doubt? If not, then I'll go to the question answer segment.
At the onset, I would like to thank you for such an elaborated and so many efforts you've made, giving us extra resource materials to read on to more, and also to Sandeep for bringing in more clarity by putting up some questions.
Yes, exactly. The best thing here in this live session is that not only me explaining these topics, but you all asking the relevant questions. So keep on asking these questions, keep on asking these questions in the forum as well. If you when you are going through the book, if you find any questions, please keep on posting and we will try to answer all these questions in these sessions. Okay, so please post your answers in the chat box. Class switching depends upon which of these?
All of these.
Yes, all of these. Yes. The D is the right answer because the switch regions I mentioned, the switch signals, the switch recombinase, which is the AID here, and the cytokine signal, right? So all of this is the correct answer. Okay, moving on to the next question.
D, differential RNA processing.
Any other answers? Please all of you try to answer. Yes, the correct answer is differential RNA processing. Somebody answered class switching, but please remember class switching is for yeah, actually alternate splicing and differential RNA processing is actually referring to the same event. Okay, acha. One more thing is class switching is for the production of several classes of antibodies and not the either whether they will be transmembrane or whether they will be secreted, right? So the correct answer is differential RNA processing, or as you may say, alternative RNA splicing. Okay, that is the correct answer. Moving on to the next question. What will be the answer?
C, the variable region of the heavy chain remains the same, but its constant region changes.
Okay, let's see. So here people are clearly divided. Some are answering B, some are answering C, some are answering A. Okay, so the correct answer is C. And why not B? Look at it carefully. The C, option C says the variable region of the heavy chain remains the same, but its constant region changes. Whereas in case of the option B, it was that the variable region of the light chain. It was just the difference between option B and C was that in case of option B it was light chain, and in case of option C it was heavy chain. So remember, the constant region of the heavy chain is only responsible for the several classes of antibodies, right? So let's go back to that slide once more. Here, look at it. Only the heavy chain, right? And not the light chain constant region. Alright? So that is why the correct answer is that the variable region, this particular region, remains the same because the VDJ recombination has already taken place. Now this particular region will remain same, but there will be differences in the constant region. Okay, there will be differences in the constant region of the heavy chain, which is why the option C will be correct. Okay, not option B. Okay, moving on to the next question. Which of the following immunoglobulins are derived from the same pre-mRNA transcript? Yes. So the answer is B, IgM and IgD. Right? The same pre-mRNA transcript is spliced differently to produce either IgM or IgD. Alright? Solution is B. And the last question.
Ma'am, can we discuss the previous question once?
Yeah. I'll just show you the slide once. See? This IgD.
I understood that the B option is correct, but what is wrong in the A option that says G and A? Because G, E, and A are also there in the single transcript.
No. Remember this, that they are not. So here you will see that all these constant regions are on the same mRNA only, but in the pre-mRNA transcript, right? In the pre-mRNA transcript, which is this primary transcript mRNA, not all these constant regions are expressed together. So I told you, when there is class switching, there will be several cytokine induced signals which will help in the expression of either IgA or IgG or IgD. Oh, sorry, not not this one, right? Either IgG or IgA or IgE. Okay, so they will only be expressed or they will only be transcribed when there is proper cytokine signal, right? But only IgD and IgM, they are only expressed together always. Okay, so in this
Thank you so much.
so in this primary mRNA transcript, you can see that only this mu region and the delta regions are being C mu and C delta regions are being expressed, but the other classes of antibodies will be expressed only when there is will be only transcribed only when there is proper cytokine induced signals. All right. Okay, so moving on to the last question for today.
A, MHC class 1.
Yes. A is the correct answer. Okay. All right. So thank you so much for attending today's session, and I feel very delighted to answer all of your queries and discuss all these things with you. If you have any other question, which edition of the Genways Immunology? I think I have the 9th edition, yes. So you can go on with the 9th edition. If you have any latest editions also, you can follow. Alright, thank you so much for joining today. Next Tuesday, we will again meet for another session, okay, with some other concepts and other queries and other problems of immunology. So see you then. Bye. Take care.
Thank you.
Thank you so much, ma'am.
Thank you.